Hello to you later on as well. Um, my name is Leah Bendlin. I'll be your teacher tonight. Um, and hopefully you're new to fungal microscopy because this is for you. Thank you for treating yourself to something new. And with that, I guess we'll get started. If you have any questions, feel free to pop them in the chat. I'm going to keep the chat window open so I can answer them as they come in. And if you want to unmute or turn your video on at the end, if you feel like it, then you can ask questions more personally if you would like to. Okay. Okay. We're pretty good here. I just need to get the chat as well. There we go. Lovely. Okay. Intro to fungal microscopy. Let's do it. Okay, so why in the heck are you even going to use a microscope? If you have something that looks really ambiguous and you have no idea what it might be, it should be fairly clear once you look at it under the microscope. There are very distinctive features that can help you rule out just a ton of things. There are a lot of species that can look very similar to the naked eye that will have very different microscopic features. So sorting out species that look very similar. If you are going to make a new species discovery, you're definitely going to want to note the microscopic features that are going on with it. And this actually happens more often than you would think because, especially in North America, the mushrooms have European names because European people were cooler with mushrooms in general. And so a lot of the North American species just, there's a species that looks like that in Europe. They'd assumed that it was the same. Turns out that it is actually genetically different than the one that's in Europe. And so these things are getting renamed all the time. And one of the things that are different with them will be the microscopic features. So if you happen to find something and you get the DNA done on it, turns out it's completely something new, you're going to need to make all of these notes about what the microscopic features are, as many of them as possible, so that somebody doesn't have to run the DNA the next time to know that this is compared to what you found. Also, it's just cool. I think it really can help to give you an appreciation for the complexity of life and all of the things that work together. You know, there's just a tinier and tinier and tinier world the more that you're able to look at it. And it's incredible, you know, seeing little, you know, amoeba-like things and nematodes and spores. So yeah, hopefully you're already here because you think it's cool, but I'll talk more about why it is. Okay, so here's an example of that first situation where you have a whole bunch of different species that basically look the same, but you know, in theory, each one of these little brown mushrooms that I'm showing you is a different species within the genus Galerina. However, if you put each one of these things under the microscope, you're going to find that they look quite different. Okay, so here are some micro photos of the actual mushrooms that were in the last slide. And the features that we're looking at here are called the chylocystidia. They are cells that are right at the edge of the gill. So the one in the upper right, these cells right here that are like long sticks with a little circle on the end, that's chylocystidia. And each one of these over to the left, it's zoomed in farther, that look kind of like bowling pins. Those are also chylocystidia, or singular chylocystidium. So you can see that those two things look quite different. So you're going to be able to compare them microscopically pretty easily. Here's an example of something where I had no idea what it was. So generally, you have two major groups of mushrooms that you're working with. They are going to be either basidiomycetes or ascomycetes. And basidiomycetes are like your gilled and pored mushrooms and also jelly fungi, in most of the jelly fungi anyway. And then you also have ascomycetes, which are cup-shaped mushrooms, and things like morels as well. So morels, basically just a bunch of cups on a stick. Um, and this thing has characteristics of both, right? It's kind of gelatinous and it also has a cup-shaped thing to it. So I really didn't know where to begin to even try and key this thing out. And when I found it and put it under the microscope, I found that there were no asci, and we'll talk about what those are in a little bit, but that's a feature of ascomycetes, or the cup fungi. So I knew that it wasn't one of those. And I was eventually able to identify it as something called Guaipiniopsis buccina, which is a jelly fungus. And here's an example of something unexpected that I found under the microscope. This is a nematode, so just like a little worm-like thing that happened to be floating around in the mushroom flesh. And I mean, how many of these things have you eaten when you're cooking up mushrooms for the first time? Probably a lot. There's a lot of wildlife happening in wild mushrooms, but they're microscopic, so you're cooking them. It's extra protein. But they're beautiful and they're doing stuff in there. Okay, so there are two major types of microscope. And oh, got someone else joining here. Oh. So the first one is a stereo microscope, and the other type is the compound microscope. We are mostly going to be talking about using the compound microscope. So the stereo microscope is a lower magnification. I think mine goes up to 40 times magnification, so that's quite low. It's lit from above, so it just has a light that goes down. And it's for viewing solid objects in 3D. So you actually get a 3D kind of view from it. And it's especially useful for when you are cutting pieces off your piece of mushroom and mounting slides, making sure that you're getting really exact cuts where you want to go. Or if you are interested in doing like slime mold microscopy. Slime molds are not fungi, but they are a passion of mine as well. And they are really, really, really tiny. And so it can be useful to just look at what are you even grabbing at, because it can be hard to see with the naked eye. That said, you don't need a stereo microscope to work with the compound microscope. They're just really fun to have. And also if you're looking at things like mosses or the textures of rocks or things like that, it can just be really neat to see the texture of the outside of things. Okay, and here is the compound microscope. This is at much higher magnifications than the stereo microscope. Mine goes up to 1,000 times magnification. And the light comes from below and it passes through the glass slide, which will go right here. And it produces a 2D image from that slide. So you don't get the 3D image like you do in the stereoscope. Everything is in one plane. Okay. And for the people who just arrived, I do have a chat window open. If you have any questions while we're going along, feel free to type them into the chat. Okay, so here are the parts of that compound microscope. So from the top, you've got ocular lenses. So that's just the thing that you put your eyeballs up against. You can take these out very easily. Just grab one and put in a different magnification. So I have ones that came with my microscope that are 10 times and ones that are 20 times. I only use the 10x ocular lenses, but we'll talk more about that in a minute. This spins around the revolving nose piece. So if you've got a friend next to you and you want to kind of point it in their direction so they can see what you're seeing, that's good. Or I store mine with the eyepieces backwards so that the dust cover goes on it better. always use your dust cover. You don't want little bits of dust to be getting all up in these precision lenses. Carrying handle, that's where you grab it if you need to take it somewhere. And here are your objective lenses. Okay, so these are the ones that really do the high-powered lifting when it comes to doing the magnifying and really get it very magnified. Here is the slide holder. So this is a little arm that moves to hold your slide in place against the other part of it right here, and it keeps it from moving around. On this diagram, there isn't a stage control, but there are little levers that can move the stage back and forth to get your specimen into place on the stage, and the stage is just what holds the slide. The condenser aperture lever is a little bit hard to describe, but it's like the iris of an eye. It controls the intensity of the light and the size of the beam of light reaching the specimen. And you can adjust it at different magnifications to control the contrast and the clarity. So just play with it as you go along under different magnifications and see what looks clearer and better by moving the lever around. And then the condenser is below that works in tandem with that. The light is what passes the light through the specimen from below. And then you have your coarse adjustment and fine adjustment knobs. They do the same thing. Coarse adjustment just does it much faster, and fine adjustment just does it more slowly. So that is going to bring your specimen into focus and out of focus by moving the plane at which your specimen sits up and down. And then there is the light intensity control. So that is just going to make the light that comes out brighter or darker, which you might want if it's, if you turn this thing on and it's like blinding your eyes because it's too bright, then you can turn it down a little bit. And there are situations in which, along with the condenser aperture lever, you're going to want to adjust the brightness. Okay. And along with that, I'd like to say, like, you don't have to be perfect at this when you start. Play with things and see what happens. You're going to have to do a lot of trial and error when you're first starting out to get comfortable with things, and that's okay. And there's also lots of different ways to do things. There's not necessarily one right way to do everything, and there's a lot of different techniques to get good results. Okay, so your magnification is something that you're going to need to know, especially if you're doing any measurements of things that you're looking at under the microscope. So to know what your magnification is, you need to multiply the objective lens magnification. So like I said, I use 10 times 10x eyepieces, and then multiplied so the eyepiece, I do use 10x lenses. And then the objective lenses, which we'll go back, those are these ones, the heavyweights that do the real magnifying. And mine are 4 times, 10 times, 40 times, and 100 times. So I am multiplying each one of those things by the magnification that I'm getting from the eyepiece. So ultimately, my total magnification for each one of those objectives is going to be 40 times, 100 times, 400, or 1,000. My microscope only goes up to 1,000, and 1,000 is plenty for doing pretty much anything mushroom related. So you want to start at the very lowest magnification and work your way up. So you make sure that you have your specimen in the field of view. You're like, okay, it's at least there. And then you turn your objective lenses around, they swivel, and then lock into place your next one. It just kind of clicks into place. And then you work your way up to the highest magnification. You don't always need to go to the highest magnification. It kind of depends on what you're looking at. You're probably going to need to go up to at least 400 times magnification. But if you are making measurements of spores or other fine details, you probably will need to go up to 1,000 times magnification. And that is the oil immersion lens. And we'll talk more about that in a little bit. As far as the 20 times eyepieces, so remember I said that I have 20 times eyepieces that came with my microscope, but I don't use them. What they do is they kind of zoom in on the picture rather than providing very high quality magnification. So it's like when you pinch zoom on your phone and then you get a like really pixelated kind of grainy look from it. So most people that I know don't usually use the 20 times eyepieces, so they don't really provide a whole lot of extra detail. Okay. So the oil immersion lens is going to be the highest magnification. So on mine, it's 1,000 times magnification. So the lens itself, the objective lens is 100 times magnification, and then my eyeball lenses are 10 times magnification. So those multiplied make 1,000 times. And Elena, I'll get to your question in just a sec. And then you want to use a drop of oil on the very top of the slide, stuff called immersion oil that they will probably send you with your microscope. Otherwise, you can buy it online. So the way that the general order of the slide goes is, okay, I'm making the slide with my hand, and then you put a little drop of water or something else, some sort of liquid, and we'll talk more about other liquids that you may use. And then you put your specimen, tiny little piece of stuff, and then you put a cover slip, a little glass square that goes on the top of it. And for the rest of the magnifications, that's fine, and you don't need to do anything else with them. But if you're using the oil immersion lens, on top of that whole getup, you put a drop of oil onto the very top of the cover slip. And then that oil actually touches the oil immersion lens, and you see through the drop of oil. So you only use that for the highest magnification. You really need to clean that when you're finished because that oil gets sticky and it can muck up the lens. So every time when you're done using that, you do need to clean that with a piece of lens paper. Now I have a slide coming up that shows you what that lens paper comes in. And I use alcohol for that, or they have cleaning solutions for that as well. So you just put a little bit of it onto these wipes and then rub it across, or maybe kind of stick it up in there a little bit if needed. And you don't want to switch back to the other lower objectives afterwards or get any of the oil on them. The oil is only for the oil immersion lens, and you only do it when you're ready to make your final measurements for things or look at it only through this because you can't go back through and look at things through the oil after it's been mounted that way. Okay, so Elena asks, what should we budget for a decent microscope? So I bought the Amscope B120C, and I'm gonna write that into the chat. B120C. And that is the cheapest microscope, or even the Amscope B100S, I think it is, or MS. I'm gonna write this in the chat too. Amscope B100 BMS or something like that. Is the very cheapest thing that you can get that will work very well for this. I spent, I think, about $200 on it. There, some places are getting better and have other things that are that cheap. I don't know of other brands that are doing it that cheap for offhand. Swift is another company that makes really good ones that are pretty cheap. If you're willing to spend more money than that, you can get better quality objectives that are going to give you a crisper picture. But the Amscope that I have is just fine for doing everything that I have needed it to do. So I haven't needed to spend more money than that. Okay. Okay, and here's like the base stuff that you are definitely going to need. You're welcome. That you're definitely going to need for doing some good fungal microscopy. Okay, so obviously you need your slides. So this is full of slides that are about this long. Here is a box that has cover slips, so they're little glass pieces that go on the top. And here's your lens cleaning paper. So it's very fine grain paper that isn't going to leave little bits of fiber on your objectives or on your slides. This is water, distilled water, and that's what I mount most things in. There are reasons to mount your specimen in other things, such as KOH, which is also known as potassium hydroxide. You can rehydrate specimens with potassium hydroxide, but you can also use water or alcohol to do that. I am going to send out a link after this, and also y'all will get the slides for this, where I'll include this link. There is a very thorough guide to fungal microscopy from another person who's mysterious, who goes by M No Line, that I really recommend, and it talks in detail about what you use each one of the stains or chemicals for. But a lot of the time, especially just when you're first starting out, just use distilled water, or I even use tap water, as long as you're not in a place that has like a ton of minerals or weird stuff in the water. Probably tap water is fine. Okay, here's your alcohol. Alcohol is for cleaning off the immersion, the oil immersion after you are done using that 1,000 times magnification, your highest magnification. 99% or 70% will work. Yes, 70% will work, and it probably is a better idea from my understanding, because it might be less likely to like eat away at things in your microscope. I mean, I use the 99 and it seems fine. I haven't had anything fall apart on me yet, but 70% is what is generally recommended, I think. Okay, so here's your immersion oil. It's kind of looks like olive oil. I don't know exactly what it is, but it's the sticky stuff that's what you use for the immersion oil lens. Okay, razor blades. You are going to need those to make very thin sections of mushrooms. Yep, you kind of know a way getting around that. You're going to need razor blades. Tiny tweezers. The finer, the better. So you could even get finer than the ones that I have here for taking just little bits of gill edges off, or if you're working with cup fungi, use it to take just a tiny bit out of the center, and just picking at things generally to get tiny samples. This little thing off to the left here is a cover for those tweezers so that if you drop them, it doesn't ruin the end. And then in front here, I have just a pencil with a pin stuck in the end of it. That's really useful for just poking at things. Let's say you're grabbing a piece of gill off of a mushroom and then it's kind of stuck to your tweezers. You can use this to scrape it off and put it onto your slide. Here is my phone. I use the phone for taking pictures so that I can share with the world what I'm seeing. And I just hold that up to the eyepiece, and then you kind of have to follow the circle of light that you'll see through it until you can get the picture clear into your phone. You can do it, but it just takes a little bit of practice. I like to go low tech and as cheap as possible, so I don't have any like fancy camera adapter that that attaches to my microscope, but some people go that route, and I'll talk more about that in a little bit. I have no idea what brand my tweezers are. I just searched around for fine-tipped tweezers or forceps, but I can look at that and maybe get back to you. Making a note. Okay, and some of these are really darn expensive because they get super, super fine tipped. Um, these were somewhere in between like regular tweezers and really fine tweezers, and they're fine. I haven't needed especially fine tipped tweezers. Like you could even use the same ones that you have in the bathroom for plucking your eyebrows or whatever. But it really helps to have finer tips. Okay, and then you will need a calibration slide. And if you know someone who has a calibration slide, you're probably only going to need it once. And that is for um, calibrating measurements on your microscope. I will talk in a minute about what that means. Okay, so generally mounting a slide, here's what it looks like. You always want to use a drop of water or other liquid. Remember, I talked a little bit about using the potassium hydroxide. You might also use a stain of some sort. Using stains is a little bit more advanced, and there are various ways to do it. You can either just have, for some stains, just putting the drop on there and putting the stuff, your fungal material into the stain is fine. For some things, you want to actually stain the fungal material with the stain first, and then soak it all up with a paper towel or something like that, and then put water onto the slide so that you're not getting too much concentration of dye. Another way to do that is by you mount your you do a regular mount where you just have your specimen in water, and then you can put a little bit of stain on one side of the cover glass, not on the cover glass, but just to the side of it. And then you have a napkin or something or a piece of lens cleaning paper on the other side, and it actually pulls the stain through. So some of that dye then gets on the tissues. But again, if you're just starting out, water is fine most of the time. You want to use the smallest amount of material possible. So if you use too much, your specimen is going to be very thick. There will be a lot of cells sitting on top of each other. And it's going to be hard to see through and make out what's going on. So using the smallest amount of material as possible is a good idea so that you can actually see the cells. And again, yeah, the stains can be used to help show translucent features, and the document that I'm going to link will talk about which stains you might want to use for looking at particular features. So if you have a guide and you're like, well, it could be this or it could be this, I know it's at least in this genus, but I'm not sure which species it is. Your key might tell you that a certain type of fungal tissue turns a color with the application of this chemical, or the specific tissue turns purple with the application of Melzer's reagent or whatever like that. So you kind of want to have your description of something to compare to so that you know what maybe you would be looking for. Just a quick note about using potassium hydroxide. So potassium hydroxide, it's caustic. It can kind of like burn your skin. So be careful with it. It can do things like clearing out material that isn't fungal related. So it can kind of make your slide cleaner. It also can do things to the cells of the mushroom that are taxonomically significant. So you don't necessarily want to mount everything in potassium hydroxide because there are features that could disappear or explode with the potassium hydroxide. That doesn't happen for most features, but there are cases in which it would affect what you're looking at in a way that might change your perception of the ID. Okay, so you've got your slide, your wet stuff, water, potassium hydroxide or something else, your specimen, and then your cover slip. Cover slip really helps to like make the wet stuff, make everything all in one plane. Okay. Okay, and then there are different types of mounts. So there is a regular mount and a crush mount. Crush mount is also known as the squash mount. So you want to use a like regular mount or non-crushing mount when you want to view the features as they relate to each other in space. So if you want to, for example, see the cells that are at the very top of the cap, the pileus, you want to know where the very top of the cap is. So you want to make sure you're not getting some other part of it. So you kind of need to see the whole shape of what you're looking at to know what is the top. And okay, so then you've got the crush mount. So you put your piece of material on there and then you use like pencil eraser or something like that and squash the cover slip down a little bit. And that actually squashes your specimen and can separate the features out so you can look at things more individually, but you don't necessarily see how they are related to each other because the pieces of them can kind of go flying in different directions. Okay, and most of the time I do use a crush mount because I'm just looking at things like spores, or maybe I want to look at the shape of an ascus, and I don't necessarily want to see all of the things as they relate to each other. Yeah, I would say looking at all of those things as they relate to each other is more of an advanced thing, and crush mount is more for doing very basic things and looking at individual features. Okay. Okay, so you are definitely going to want to take pictures. It's fun to share with people what you're doing, and it's also good for science to see what features you use so that you're recording, hey, this is what I saw under the microscope, and here's the proof. And if someone wants to look at it later and say, oh, you misinterpreted that, there's some data about why. So yeah, like I said, I use my cell phone. You can also use a digital eyepiece or USB camera. So these are things you can buy in addition to your microscope that will go in place of the eyepiece. I bought one of those when I was first starting out. It was a USB camera and the quality of it really sucked. So I would definitely recommend looking at the reviews for these things before you go buying a USB camera. And I have heard similar reviews from other people who have bought USB cameras that they are generally not very good quality. So make sure you look for those reviews. HDMI cameras are often more highly rated. Or you can just have like a regular camera if you have some sort of fancy camera, or even your cell phone, you can get mounts for the microscope that will keep your chosen camera at a consistent distance away from the eyepiece. So then this is going to come into play in a minute, but if you're using a program to measure things, you need the distance of your camera to always be at a consistent length away from the eyepiece. Does that make sense? Okay, so here's the calibration slide. In real life, this is a clear piece of glass, and you will probably only need it once. This is what it looks like in real life, and below is what this tiny bar that you can barely see looks like under the microscope. So this is a given known distance. The calibration slide does not come with your microscope. You will have to buy it separately or borrow one from somebody, at least at the one I got. So there is, and also if you just like want to start out and just look at spores and their shape and you don't really care about measuring stuff, you don't need to have one of these right away, but you will probably quickly find that it is important for you to have this because you'll want to measure things. And also, most microscopes do have about the same measurement scale. So chances are, when you're looking at 1,000 times magnification, each one of these units is going to be 1 micron. So that's the unit of measure. However, it's good to get this slide to make sure that your microscope is in line with that. So what you do is you've got this slide and then you look at it under each one of your magnifications and count how many of these units align with a reticle. Okay, so a reticle is a little piece that pops out of the eyepiece, and there is a little ruler inside of the eyepiece. So you'll see both this on under your eye, and you will also see another ruler on top of it. So you're going to measure how many of these are present for each marking on your ruler in your eyepiece. Okay, and then write that down for each one of those. And again, the document that I'm going to link tells you more details about that. But typically, when you're looking at 1,000 times, each one of the divisions in the ruler in your eyepiece, the reticle, is going to be one of these divisions. So it'll be 1 to 1 when you're looking at 1,000 times. Okay, so once you have that calibrated, you never need to use this slide again, probably, unless something weird happens with your microscope and it gets super old and messed up and you want to see, but usually you don't need to do that anymore. And then you can just use the little piece in the eyepiece and measure with that. But at different magnifications, things are going to be different sizes. So when I'm looking at 400 times magnification, it's not a 1-to-1 ratio with that, with what I'm seeing in the little eyepiece. It's at 400 times magnification. I need to multiply my measurements by 2.5 to get the actual real life measurement, right? Because the 1-to-1 at 1,000 times, 400 times 2.5 equals 1,000. Does that make sense? It's a little bit complicated, but there are guides to doing this online if you need that. So I usually just measure manually with my reticle, so that little ruler in the eyepiece. And I pop it out when I'm not looking at it and just use a regular eyepiece that doesn't have a ruler in it because I don't want that kind of in my field of view. Yeah, you probably will have to buy a reticle separately, AJ. Mine did not come with one as well. I had to buy one separately. Some of them may come with them, but yes, you will need that. And then you can also measure electronically with a program such as Piximeter. And Piximeter is a free downloadable program. The website is in French, and I forget what the French word is for download, but you can just use Google Translate to translate the website. And after that, the program is in English. Le download. It's not le download. Sorry, but that's good. Okay. So here's what it looks like when you're looking through the eyepiece the reticle that has the little ruler in it, okay? So if I was looking at this and I know that I'm looking at it under 1,000 times magnification, and here's the spore, this spore is about 10 microns wide. So that's your measurement for that. And if you are measuring for science, you're supposed to measure around 30 spores because they can vary a little bit in size. And you're also supposed to drop the very highest and lowest ones because they're weird outliers. I usually measure like 10 or so. It's tedious to measure a lot of them, but it depends on how much you care about your specimen. Okay, and here is a screenshot from Piximeter. And the way that this works is once you have it calibrated to your microscope, so it knows your standard magnifications or what you do is you just click on each side of the spore or whatever the other feature is, and it calculates that distance for you. So it can be really handy if you have the setup that has a camera that stays at a given distance, but I do not, I just use my cell phone. And again, we're going to be sending out these slides, and so that'll include the link for this, but you can also just Google Pixie-meter. Okay, so now we're going to talk about some of the things that we actually see when we're looking under the microscope. So basidiomycetes are a major group of mushrooms that include things that have gills, things that have pores, like your boletes and polypores, and also the coral mushrooms, and jellies as well. Most jellies are also basidiomycetes. And even though these things can look very different, they are related to each other in some fundamental ways. Okay, and kind of the gold standard for looking at gilled mushrooms under the microscope is the Roman aqueduct section. And you could see why it's called that, right? And here's what it looks like in real life. You take a slice through this cap here, and you know, all of the arms of that aqueduct are the gills of the mushroom. I have to say that these sections here are not going to be thin enough. You do need a thinner section than that, or there's just going to be too much fungal tissue, too many cells sitting on top of each other to see things very well. It takes a lot of practice. I did learn a new technique recently that I think is going to work pretty well. And I put it in my Instagram stories today if anyone wants to look at it. And I also am keeping it in the stories under microscopy in the highlights. But the basic way that it works is that you have two razor blades without any bevels on them, just like the razor blade that's in this photo. And you wrap a piece of tape around, like a piece of clear tape around this, oops, sorry, this part of the razor blade, the unsharp part, okay? All the way around it like that, leaving the bottom part open. And then you take another razor blade and stack it on top of that, and then wrap it around the entire two razor blades so that there is, you know, a tape's width of space between the two razor blades. And then you cut down on your mushroom, and hopefully your section is between the two razor blades is going to be thin enough for your slide. And I used a little pin to kind of pry it out from in between the razor blades. So I just tried that today for the first time. I felt like it worked pretty well and I would recommend it. Okay, so when you've got your slice, here's some stuff that you're gonna find underneath there. So here is, if you had this Roman aqueduct section, here's what you would see. The pilipellis is the top of the cap, so we're oriented in space, and here's the gills hanging down. And you're going to see lots of little things going on. All of these little bumps here without thingies at the end are called basidioles, and they are probably immature basidia. Basidia are the spore-bearing structures of basidiomycetes, and they are club-shaped structures. Okay, so if we look on the left, this is a basidium, zoomed in. So it's this club-shaped structure, and it has what are called sterigmata on the end of them. So they are just little stalk kind of things that hold the spores. And if they are spores on a basidiomycete, they're called basidiospores. Okay, so each one of these, we're zooming out now. Here are some of the sterigmata and little spores that are on the end of those mature basidia. And then there are specialized cells that are at the edge of the gill called chylocystidia. So chylo means margin, so they're at the very margin of the gill. And they can look a lot of different ways. Remember, one of the first slides showed some things that looked like sticks with circles on the end, and there were bowling pin-shaped things. So those are Chyloscystidia. They can look a lot of different ways. And if you have a good resource that you're comparing to, it's going to tell you what those Chyloscystidia should look like. And there's a lot of crazy words for them that you're going to have to look up. And then there are Pleurocystidia. So Pleurocystidia are on the face of the gill. So you're going to find them on the side and up in here. They may look the same as the Chyloscystidia, or they may be different. Your mushroom may have Pleurocystidia, but not Chyloscystidia, or the other way around, or it may have no sort of special cystidia. It may just have basidioles and basidia. So these are features to look for. If you're looking at the stipe of the mushroom, you might also see Caulocystidia. So there's just a bunch of weird cells that nobody really knows what they do, but they look different in different species, so they can be helpful as diagnostic tools for ID. If you get a little more advanced, there's also the orientation of cells that are in the flesh of this sort of area can help you with ID as well. Okay, any questions on that? Okay, I don't see any, so hopefully we're good. Okay, so this is an Inocybe. So you might recognize Inocybe, the genus. It means Ino is fiber and Cybe means head. So there's a lot of mushroom genus names that are something Cybe because mushrooms look like heads. And when you get it under the microscope, you will see all of these little projections that are coming out, and there are encrustations on the end. So like little dark grains of sand kind of stuck to the end. And that is very typical for Inocybe cystidia, but not for most other mushrooms. So if you think it's an Inocybe and you see these under the microscope, you know, that's a good confirmation that that's what you have. Okay, and here are Chyloscystidia of a Flammulaster. And remember that Chyloscystidia are the ones that are right at the edge of the gill, on the bottom of the gill. And these ones are more kind of bowling pin shaped. Oh, okay, yep, I'm moving back to the drawing. Is this what you wanted, Elena? And also we're gonna send out these slides too. If there's anything that you missed, you'll have a copy of it later. The Pleurocystidia or the Chyloscystidia? So Pleurocystidia are here on the side of the gill, and Chyloscystidia are at the very end of the gill. Does that make sense, or are you missing something? Okay. Yep. Okay, so these Inocybe cystidia are going to be, this doesn't look like it's the end of the gill, so this is probably Pleurocystidia, so they're from the face or the side of the gill. But there are cystidia that look like this all over Inocybes. Okay, and these are the Chyloscystidia, which are going to be right at the end of the gill. This note also the pink color of this. There's a stain that's been applied to this so that you can see the cells better, because otherwise they'd probably just be kind of clear. Here's another type of Chyloscystidia. These ones I think look like cow udders on steroids. They have all of these like pokey projections going out. I'm from Wisconsin, so that is just what I thought of. And this, again, this has been stained with something, probably Congo red. That's a very common stain to help the features show up. And here are Chyloscystidia of a Pluteus. And these ones look like little horns. And Pluteus is also known as the deer mushroom. And the reason it's called the deer mushroom is that it has these little features that looked to someone like deer horns. So even though it's kind of a brown deer colored mushroom, the reason it's actually called that is because of the cystidia that have these horn shaped things coming out. And you might note that this also looks fairly similar to basidia. So the basidia can be kind of shaped like they usually don't have this kind of thin neck at the end, but I guess I just want to note that cystidia can sometimes look like basidia. Some cystidia are kind of rounded. The size of them is the one way that you can help to differentiate between the two. Often the cystidia are going to be larger, but hopefully that's in your description. Like they're this long and the basidia are this long, so they're hanging way out farther than the basidia are. Okay, so that's going to cover it for now for basidiomycetes. And then there's my favorite group, the ascomycetes, the cup fungi. So this includes things that are disc-shaped or cup-shaped, and it also includes things like morels, but basically morels are a whole bunch of little cups with a stipe. And it includes things like Helvella and Geometra. And the spores on these things are coming from the inside of the cup. So whereas with your basidiomycetes, you're going to usually take samples from the underside of the cap, for the cup fungi, you're going to take it from the top if it's a disc-shaped thing, or from the inside of this cup, or from the outside of the wrinkly flesh on something like this. So not from the underside. And if you're doing a spore print, for example, with something like this, you just lay it down on whatever you're collecting the spores on. You don't have to separate the cap from it because the spores are coming from the outside. And just a note on when you're measuring spores, it's very important when you're measuring them to get mature spores. So if at all possible, you should try and do a spore print onto a glass slide and then scrape a little bit of those spores up from the glass slide and put it onto your sample, so that you're making sure you're only measuring mature spores and not immature spores, which might be smaller. Okay, so here's the main thing that I look for when I am looking at ascomycetes. So this is me taking a little section of the interior part of the cup with my tweezers, very small section, and you see these test tube shaped structures called asci, or singular is one ascus. And then the things inside are the spores, ascospores if it's on an ascomycete. And typically they're going to look something like this. They are often oval shaped and there are usually 8 of them, although there are variations on that. And then there are paraphyses, and paraphyses are these elongated kind of spacer cells that are in between the asci. I don't think anybody really knows what they do, but they're sterile. They don't have any spores on them, and they can look lots of different ways. They can have all of these little kind of grainy things inside. They can be different colors. They can be different shapes. And especially like what the tips of them look like can be very important. So here is what I'm seeing under the microscope when I'm looking in real life. So this is a Scutellaria, and if y'all are familiar with that, it's a little red-orange cup fungus that has kind of eyelash hairs around the outside of it. And here are the spores inside of the ascus, and here are all the paraphyses, and they are filled with kind of an orangey content. This is just mounted in water, so all of that color is actually coming from the mushroom itself. Okay, and this is the same thing, but just zooming in a bit. And you can see that the paraphyses are kind of thickened at the end, so that's important. And there is also some texture that's going on the outside of the spores. And if we zoomed in even more to 1,000 times, we might see that it has like little, little kind of warty things on it, or reticulation. So that's kind of a fishnet raised pattern. Or they might have a cell wall around them that's thicker than the rest of it. And all of these things can be useful for diagnostic ID purposes. Okay, so this is Pseudoplectania melena. It is a small blackish cup that's fruiting right now in the Pacific Northwest. And it has these really distinct paraphyses that are hooked or sometimes even coiled, and that's pretty unusual. And that specific kind of paraphyses that are hooked like that are called croziers. It's like a shepherd's hook. And also notice that these ones have round spores instead of elliptical spores. And there are several other black cups that look similar, but have elliptical spores instead. So that will easily help you rule out half of the things that it can be. So most of what I think you probably are going to want to do when you're just starting out is to look at spores, because a lot of the time you can just get to your ID only by looking at the spores and measuring the spores. They come in a lot of different shapes and sizes. Like I said, for ascomycetes, they are often oval or just round, but basidiomycetes especially have a lot more variation. And like I said, make sure to only look at mature spores when you're measuring them. Okay, here are paniolus spores, probably at 1,000 times magnification. And you can see that there is a little bit of a kind of opening here at the end of the spores, and that's called a germ pore. And that's where the spores would basically sprout new mycelium. And not all spores are going to have a germ pore, but there are several genera that do, so that's something that can help you to identify them. Here is an ascomycete that has gutules. So these little oil drops that are inside, or sometimes your description will just call them drops, whether or not the spores have those at maturity and how many of them and where they're placed in the spore can be important for ID. So this one, they more or less, most of them have two. Some of them have a little bit more than that, but that's important. The function of the gutules seems like is to feed the spore while it's growing, so it's like a little fatty deposit. Okay, and then some of them have just wild shapes. This one's kind of star-shaped, so Asterophora is the genus for this, and that I think that means like star-bearing. So this mushroom is bearing little stars. And here's an example of an amyloid reaction. So an amyloid reaction is with the application of Melzer's reagent. So you would mount your specimen in this chemical called Melzer's reagent. And it can either turn features this kind of bluish black color, or it can turn them an orangey reddish color. And the orangey reddish color is called a dextrinoid reaction. Or it can have no reaction and they just stay clear or whatever color they were before. But color changes with the application of Melzer's reagent are significant. This is a little bit tricky because Melzer's reagent is basically impossible to get within the United States because it is a controlled substance. It contains a material that is used for making date rape drugs, so it's a controlled substance. However, it is really useful for mycology. You can use something called Lugol solution instead that you can get here, but it doesn't work for everything. I am going to link at the end of this presentation a German website called myco-service.de that can sell you and ship you Melzer's reagent. AJ asks, are there mounting liquids that support a spore sprouting while allowing for visibility under the scope? You mean like, are you worried about like killing the spore with a liquid? Is that your question? Yeah, okay. So I would think that water would be good to not kill it, but basically like you're working with such a tiny amount of material when you have this slide. I mean, you don't undo the cover slip and then take the spore out and then put it outside or whatever. So, I mean, usually you just throw the slides away when you're done or wash them. So I don't know, that's not something I've really thought about before. Oh, to observe a spore sprouting. So, I mean, that happens so infrequently. Like it takes a while for it to happen. So I actually have seen spores that were sprouting um, underneath the microscope, but maybe only like once. You can see a little piece of kind of hyphae that are coming out of it. But you can't like watch it in real time because it takes too long. Okay. So does that, I don't know that anything that you mount it in would make it so that you can't see the hyphae coming out. But that's just not something that you see that often. Cool. Okay, so here is an example of what a dextrinoid reaction looks like. So remember that is the different kind of reaction that you see in Melzer's reagent. Okay, so you have this blue-black reaction, and then you have these red-orange reactions. Okay, and here is a wild-looking spore from Trichoglossum. So these are like club-shaped fungi or earth tongues, little black things that come out of the ground and then have a black tongue shape at the end. And they have these really long, super narrow spores, which is unusual. And the first time that I saw something like this under the microscope, it was much shorter, but it had these little sections that were in it. And I was like, what's up with those? Are each one, is this like 4 spores that are stuck together? But it's not. This whole thing is just one spore, and then it is many septate. So each one of these divisions in here is a septa. So if your spore is septate, that's something to note, and it's going to help you with your ID. Okay, and I will take questions, but I do really want to quick show you a video that I have of me mounting a slide, so you just get the process of what it looks like in real life. So just a moment while I get this up here. Sound. Hi, so we're gonna do some microscopy today. I just did a fungal microscopy class online, but that's kind of just me talking at you, and it's definitely easier if you can see the process of what we're actually doing. So I'm gonna show you what I'm doing. Here I have a spore print. This is from an agaricus that I did on a slide, and I'm gonna put some of those spores under the microscope. Okay. So first I've got water, and I put a drop of water on the slide. Okay, then I'm going to use my little doohickey and scrape some spores. And I can see that I've got some on the drop. Okay, and then I'm going to get out a cover slip, trying not to touch it too much with my hand. Just one. They look a little dirty, so I'm going to clean it with my shirt a little. It's probably fine. And then I place it on here. Okay, and if there was more material on this, like a bit of mushroom flesh, I might squash it a little bit by using the end of the pencil, not my finger, so I don't get my finger grease on it. Okay. And then I'm going to turn on my microscope. And there's this little arm here. And I'm going to put it inside here. It gets held with the edge, and the arm goes into place. And then I use this thingy right here to adjust where the slide is. And I make sure that it goes so that it's lined up with the objective here. And then I'm going to look through
and see if I can see anything. And I'm using the coarse adjustment knob here first. There we go, until something comes into focus. And then maybe adjust a little bit more as needed. Okay, so I know that I at least have my specimen in focus now. Something is on there. And then I'm going to go to a higher magnification, so it's zooming in more. And then I'm using my fine focus again to zoom in more. Okay, so now I have my spores on the screen. I've got tons and tons of spores. This is great. Okay, but I am on the 10x objective, and I have 10x eyepieces on here. So I'm at 100 times magnification right now, and then I'm going to switch to the 40 times magnification, and that's times 10 of 10 and my eyepiece. So these are 10x objectives. So now I'm looking at 400 times magnification. All right, and I'm zooming in again with my fine adjustment. That's what I'm doing with my hand here. Okay, and now I've got spores. Okay, so now I would start measuring spores. And I'm gonna have you look through the screen at what I'm looking at here. And you can see their size through this. Okay, so as you can see, they're quite small. And because we're looking at them under 400x, that's not a 1-to-1 ratio with the little ruler. So I have a cheat sheet here. I'm going to show you. So when I am looking at the microscope under 40x, that is going to be 1 RU, that's a reticle unit. That's the little ruler I'm looking at through the eye. That's 2.5 microns or micrometers. That's a measurement. So for every 1 unit that I'm looking at through the eyepiece, it's going to be 2.5 microns. So if I find something and the spore is 3 reticle units long, then it's actually 7.5 units in real life. So that is how you would measure the spores. Hopefully that's helpful to people.
Okay, we're back. And I'm ready to take questions if people have them. Oh, hang on. There we go. And where's my Zoom? There we are. Hi, everyone. And if you want to turn your video on, if you want to ask a question, feel free. It's nice to see people's faces if you want. Maybe we don't have any questions. Maybe we covered everything.
I can ask a question.
Okay.
If you could only pick one thing, and this might not work, what would be more important, spore size or shape on the basidiomycetes?
Size, but it really depends on what species you're looking at. Usually microscopy is going to be most useful to you when you already have some idea of where to place it, but you're trying to decide between different species. You know, so you know that you have an agaricus, but you have a spore range for it, and so they're going to be the same shape and color already, but one of them might be quite a bit larger than the other. However, between different genera, there can be very different shapes. And so if it's between one genus or the other, then the shape could be very important and the size not so much.
So both.
Yes, both depending on what you're trying to compare.
Okay, thank you.
Yeah.
If folks were shopping for, let's say, a microscope off of eBay, is there anything to like avoid? If you just look at magnification, are you going to be okay?
Yeah, I mean, as long as it's a newish microscope or one that they guarantee is in good condition, the only thing that I would really worry about is one that is used enough that it's dirty and requires servicing or is missing parts or something like that. But basically, as long as you're getting something that has an oil immersion lens, it goes up to 1,000 times magnification, you're probably going to be fine.
Okay.
And as long as there's a light source on it, there's like kids microscopes that only have like one objective to look through in your eyes, and they don't have any light source. I wouldn't recommend anything that doesn't have a light source.
Okay. Thank you.
You're welcome.
When you were getting started, what are some of the things that were just kind of cool at first? Like shifting from like the stuff you can touch and see with your naked eye versus like moving into this new realm.
Yeah, that nematode, that was like an early on thing that I saw, and seeing bacteria. You can see like sometimes bacteria don't look like much, but they're little like dots of stuff that are swimming around or might be like crowding around the sides of the mushroom, and they're eating them all the time. Like bacteria are everywhere. Looking at water bears.
They're on the mushrooms? Water bears?
Not usually on mushrooms. Usually I'll see them like in moss, and if I want to look for them and show them to people, usually I'll get a piece of moss and put that underneath, and it's almost without fail you can find them on moss.
Oh, cool.
So tardigrades, for people who might not be familiar with water bear, they're little creatures that are super cute. And just like the diversity of shapes of spores. And I think the thing that probably pleased me the most was being able to say for certain what things are, because you start out and you're like, oh, I totally know what it is because it's the thing in the book, and then as the longer you go, you learn that it's like not actually that certain. And when you can actually compare the microscopic features to descriptions, you can say pretty much for certain that something is what you think it is, and I like that certainty.
That's a really good point. Yeah, there's nothing to shake your confidence like getting into mushroom world and trying to ID things.
Yeah. Yep. AJ asks, where can one find good visual references for microscopic identification? Again, this guide to fungal microscopy that I am going to link you to, and it will be on one of the slides at the end of the presentation, is incredibly helpful and basically will answer every single question you might have that has to do with fungal microscopy, like bless this person who created it. Yeah, so I would recommend that. Mushroomexpert.com has a microscopy kind of guide, and Alan Rockefeller has a intro to fungal microscopy presentation that's available on YouTube as well. It doesn't go over all of the things that we covered, but it's a good primer. And then some more specialized texts. Let's see, there's, oh, okay, I'm gonna run into the other room for a second. I'll be right back, but I have a good book recommendation. Okay, so it's backwards. It says how to identify mushrooms to genus 3 microscopic features, and by David Largent, Roy Watling, and David Johnson. So this is a good one for reference.
Could you say that title one more time?
Yep. How to identify mushrooms to genus 3 microscopic features. Yeah, I heard that maybe, is it out of print? I don't know, just keep your eye on it. Okay. But that, again, most of what you will want will be included in this PDF that I'm going to send out. It is continually useful to me and it will be a great resource for you. And the PDF is also linked in my microscopy highlight section on Instagram if you use Instagram.
I've appreciated just watching your mushroom knowledge grow over the years. And I'm just curious, what's your sort of your own personal best practices? Like if you're going out in the field, like how many mushrooms you take home for IDing? Like, for example, I get overwhelmed and busy. So I'm like, I'm only going to take home one this time. What are some of the things that you do personally to grow your knowledge and to still have fun out there and to introduce and to integrate the microscopy work into your what you do?
Yeah, so great question. I do take too many things home a lot. And one of the best things that has helped my sanity with that is to just tell myself that I don't need to get to everything. Some of these are probably going to rot in the fridge and it's going to be okay. I don't need to get to everyone, but I then have the option to if I want to. And to try and only take things that I have a pretty strong interest in following through with. But realistically, a lot of the time things rot in the fridge and I open up the tackle box full of stuff and there's a pile of mushy, maggoty stuff. And I'm like, okay, well, guess I'm not doing that one.
That's good to know that I'm not alone.
Yeah, but another thing is just hanging out with people who are new to doing it and have some interest, obviously, but like the enthusiasm of people who are new to the subject and just becoming fascinated is invigorating. Any other questions? Oh, okay, I hear Gioli. I'm probably pronouncing your name wrong. Can you work from frozen samples? No. So usually you would work from a dry sample if you're going to store a sample. The reason why is if you have a fresh specimen, and you just put it into the freezer from fresh. Oh, Eileen, hi. It can burst the cells of the mushroom. So that's obviously going to affect what you're doing if the cells are burst and mangled. So what I would do is dry the mushroom and then you can rehydrate it later for microscopic use, either with water or with alcohol or with potassium hydroxide. You're welcome. And also, if you all think of questions later that you didn't think of now, feel free to write me whenever on Instagram. I'm at Leah_mycelia. And if you're local, hopefully we're going to be able to get together in person sometime within this year. I'm a member of the Oregon Mycological Society and go to every meeting there and would be happy to talk with you about stuff. Or you can email me, leahbendlin@gmail.com. Anytime I have some time, I'm happy to talk fungi with people. Oh, Elena, are you saying something? I can't hear you.
Yeah, sorry, I was on mute. Do you think using a microscope upped your ID skills with the little brown mushrooms?
Oh yeah, for sure. Yeah, because like I said, you get confirmation that a thing actually is the thing that you think it is. And so you're not just being like, yeah, it's probably this. It looks like the thing in the book. So you can see that it has that spore shape, it has that spore size. These reactions happen when you apply chemicals to it. So yes, definitely it helps with that.
That's cool. I think my last question is, what about the slime mold world captured your imagination?
They're tiny. They are beautiful. For folks, if folks use Instagram, my friend, she goes by Marin Mushrooms, Allison Pollock. Marin, MARIN _ mushrooms. She primarily photographs slime molds, and her photographs are just incredible micro worlds. And the slime molds, a lot of them when they're mature can be kind of iridescent.
Oh, wow.
Yeah, they're just gorgeous. Like there's the dog vomit slime mold that a lot of people know about that looks like dog vomit, and that one's cool, but the much smaller and less noticed ones, if you can get good photos of them, are just incredibly beautiful and diverse. So I guess I am personally very interested in things that are quite small. Obviously, I'm doing a microscopy class, but you know, there's lots of scales of tiny worlds that are fascinating to me.
Cool. Great answer. Thank you.
And thank you for the good questions. I appreciate it.
Sure.
Okay, it looks like we might be out of questions. Thank you all for sticking around, and let me know, you know, on Facebook or Instagram or email if y'all have anything else that you want to talk about. And feel free to send me your mushroom pictures and ask for help with ID all the time. I love that. Thank you for coming.
Thank you.
Thank you. Good night. Bye, everybody.